Today we will be discussing proteins in DNA replication. So what are we doing? This is DNA replication. Why do we do DNA replication? To make new cells. Where do we do DNA replication? In the nucleus of cells. And when do we do DNA replication? During interphase. Things you will need include a paper, different colored pens, and a mind ready to learn. First thing to do is to draw a diagram similar to the one displayed on the screen. This is your DNA strand with the replication fork. Do you remember the significance of the 3' and 5'? In the first step of DNA replication, helicase comes in and breaks the bonds that hold DNA together. In addition, topoisomerase attaches to each DNA strand, relieving tension caused by helicase's bond breaking. In step 2, DNA polymerase III moves across and makes new DNA. But DNA polymerase III is not very smart. Without a marker, it does not know where to start on the DNA. Therefore, RNA polymerase puts on primers, which are the markers or landing spots for DP3 to start. DNA polymerase III goes from 5' to 3' from the perspective of the new strand. This is extremely important. I repeat, it is from the perspective of the new strand, not the old strand. But wait, you must be wondering, if DNA polymerase III continues in one direction, what about the area boxed in yellow? How does it get new DNA as helicase continues to break the bonds? Well, we call the strand that has the yellow box the lagging strand, and it is rough. The other is the leading strand, which is smooth. Remember that DNA polymerase III is not very smart. It only thinks that it can go one direction. Therefore, in order to continue DNA replication on the lagging strand, another RNA polymerase needs to come in and another DNA polymerase III to make DNA on the new part. That is why it's called a lagging strand, since it lags behind the other strand, which is not impeded by having to stop and get a new DNA polymerase III and RNA polymerase. The segments on the lagging strand from the RNA polymerase are called Okazaki fragments, which is the reason why the lagging strand is rough. On the leading strand, there is not as many Okazaki fragments because there is only one RNA polymerase. For a more accurate representation of the Okazaki fragments, turn to page 303 in the AP Biology book. In the third step, DNA polymerase I replaces the RNA polymerase and places DNA there. Ligase glues the free end of the new DNA to the old DNA, making the strand complete. These fragments usually occur at the RNA polymerase sites. Does anyone have any questions? I wish you the best of luck on the test.